He is the co-founder, the CEO, and the Chief Scientific Officer of VITAS. Now, listen to this. He's authored or co-authored over 60 peer-reviewed articles. Now, that is incredible in itself. And then, since establishing VITAS in the mid-90s, he's transformed the company from an academic spin-off to now into a global leader of dried blood spot technology. So we are so honored, so excited to have him with us on this specific call here today. And I want you all to put your hands together. Here is Thomas Gundersen. The word is yours.
Thank you, Hilda. That's so nice. Not bad. 60 publications, And yesterday I had another one, so that's 61. I'll tell you about that later. Yeah. So should I just go ahead and do my presentation?
Absolutely. We would love to hear.
Okay, I will share it. So you can all see it? Yes. Perfect. Okay, so the topic for today is kind of what I already been talking about previously, but I have some small twist on the message. So I will hopefully give you some input on how you can convince your customers or coming customers that the credibility of Zinzino and VITAS as a subcontractor for Zinzino is really high. I will show you some data on the precision and the accuracy on the different assays and how we obtain this, how we validate this. And I will also put some emphasis on reassuring you that we have the capacity currently, but also that we are innovating to be prepared for the future. So, as you know, VITAS is also a pretty mature company. We were established in 1994, and we have now been in operation for 31 years. So this company is a spin-off from the University of Oslo. Actually, one of the first companies spinning off from the University of Oslo. So I did my master, I did my PhD, and I was a postdoc at the university. And I had this lab there that we slowly took into a commercial company. So I like to say that we are not like a regular startup, more like a fine wine, developing slowly with the academic thinking first, with the quality, and then making money at second or third. So our customers are different from the different region are of many sorts. We have academic institutions. Sometimes they have an idea and it turns into a small biotech company. If this biotech company is successful, it turns into a pharmaceutical company and even maybe a big pharma company. Then we have food and nutrient producers like Zinzino, and then we have hospitals and primary healthcare, and also a number of startups, small startups growing into larger companies from all over the world. So this is where we are located in the Science Park in the university campus. And this is a really nice place to work. In this building, more than 3,000 people work every day, divided into 300 companies. And they have an idea that they probably had at the university, and they are moving here, having a small office and trying to evolve this idea into a flourishing business. So, VITAS is here at the top. We have like 80% of the top floor in this building now. In both towers, it's about 1,000 square meters. And we operate as a contract research organization. So we validate, we develop, we validate, and we apply analytical methods for different purposes. So this we do for our customers. And our customers could be research organizations working with obesity and related diseases like diabetes or coronary heart diseases. They also work with dementia and Alzheimer's disease, which I think is quite important because my father recently died from dementia and Alzheimer's. We also work a lot with newborn and premature children. So this child here you see is less than half a kilo. it would fit into the palm of my hand. And children as small as this, you cannot take a regular blood sample from. So you need to prick the heel, one single drop of blood onto a paper, and then analysis. And since this is quite tricky and this is what we specialize on, we have a lot of projects related to these babies. We also work with companies that develop new drugs. So in addition to being a CEO, Hilde also mentioned that I'm the Chief Scientific Officer, meaning I'm a scientist, a researcher. I publish papers. And these are some quite recent examples of publication that I have published along with my peers. And you can see from the blue bold headings that they are related to obesity and diabetes. They are inflammation markers and dementia. There's also coronary heart diseases and biomarkers for risk stratification of this disease. So I guess you know something about scientific journals. They come in good and bad, as most things in life. And this kind of quality is described by something called an impact factor. So this is the number behind the title of these scientific journals. And I would say that a decent scientific journal would have an impact factor of 5 or more. So the higher the impact factor, the more your work is cited. So one example is the American Journal of Clinical Nutrition having 6.8. And then, for instance, Nature Communication, quite famous, 16.7, The Lancet, 44, and then the BMJ at the top with 105. So these are all scientific journals that we have published in. So the reason I'm telling you this is to kind of convince you that we are working on a really high level together with our sponsors. And quite interesting, I would say that my last publication is this one. And I think Martina will tell you more details about this later on. So this is my first publication together with Cincino. And you can see that Martina here is the first author and Colin the last author. So I think this has been really hard work, you know, been ongoing for almost 3 years. And I think it's very nice that this article now finally is online. Congratulations to you all. And on our website, you will find some nice pictures of Dag, for instance, but also a few quite very famous scientists, researchers. In the middle here, John Danesh from University of Cambridge in the UK. John is a very bright guy, very intelligent, clever with people, and the youngest professor ever at Cambridge. I don't know if you know, but Cambridge is about 1,480 years old. So this is a good guy. And to the right, we have Timothy Spector, which is a professor at King's College in London and a leader of the UK Twin Biobank, and also one of the founders of Zoe, a very famous UK company. So you can read from the testimonials here that they are quite satisfied with the services that we gave to them during our collaborations. And I would say that they actually really rarely do things like this. They post things like this on the internet using their photo. So I'm quite proud of that, actually. And this and a lot more you will find on our web pages, vitas.no. You will find a long list of publications where the employees of the company are co-authors. And you will find an even longer list of publications where we are not co-authors, but where we have done the analytical work. And if you go into the website and check kind of the length of this list, it's pretty impressive, I would say. And the reason for this is that we have been in operation for 30 years, as I said, and that we have hundreds of projects every year. And after some years, all of these are mostly published. And this kind of, I would say, showcases our experience, you know, and the diversity of the project that we have. You will also find a list of methods, application that we offer. These are developed in-house over time, validated as needed, and we use some of them every day, some every month, and some every fifth year. Currently, it's about 650 methods. And this slide shows logos of a mix of companies and Ivy League universities, biotech companies from all over the world. So these are customers of us. Some of them are present customers, some have been customers in the past, but they also kind of showcase, I would say, the diversity and the trust, I would say, from some of these really famous companies. So by this, I hope I have kind of showed you or convinced you that we have high quality, that we work with the best, that we publish really high impact scientific publications in high impact journals, that we work with really important topics like cancer development, drug development, and premature children, and that we have the scientific excellence needed to also convince your customers that we are kind of the right stuff. So in addition to all of this, we work with this company, Zinzino. This is you. And as you probably know, Zinzino is our kind of single largest customer and has been for a few years now. And I really value this collaboration, both for the business, but also for I think it gives meaning to work with Zinzino. VITAS is a really small company compared to Zinzino, but we are also working. I mean, every time I show this slide, these numbers change, you know. It's kind of funny because when I updated this slide, you know, before this presentation, that number here in red was actually quite different, you know. So that's kind of good to see. And what's also kind of typical for VITAS is that the human number here, 34, is not increasing as fast as the robotic number, you know. And this is a part of the secret, you know. The strategy is to have at least 2 robots per human, and in the future 3 or 4 robots per human. And this is like kind of secret, you know. This is why we make money. This is why we are able to keep up with the samples, not sample numbers that you send us in the same space. And also you that have visited us know that it's quite tight, you know, in the lab, and it's getting tighter and tighter, I can promise you. So this is my kind of senior team, and this is also kind of something I'm proud of, because the first 16 employees that I hired myself, you know, are still working in the company. And I think I've said something about me and the company, and it's not that bad to work in this company, which also makes me proud. And on average, they have 17 years of experience, which is really important to have as a basis, I think. And many that visit us, they kind of think that they will be welcomed by someone like this, you know, that the robotics in our lab, they look like humanoids, but they are quite boring, you know, looking like this. Hamilton machines and the GCs doing the fatty acid analysis. But these guys, you know, they are kind of the star of this company. Sorry. And then I think it's quite nice that the name of the robot is Starlet, because this guy serves these guys. Those are the GCFIDs doing the fatty acid analysis, and one robotic, one Hamilton, works together with 4 of these GCs. Just until a few months ago, they used to have the capacity of doing 560 samples per day. But then we did some innovation, you know, we changed the speed of the analysis. This is the fatty acid analysis, and the top one is what we used to do. It was also a fast GC with a cycle time of about 10 minutes. Then we innovated, we cut the time in half, you know, going from 10 to 5 minutes. And what is the result? Well, this team now do not do 520 samples, but 1120 samples with the same setup. This is quite known, you know, no additional cost, just double capacity in the same space, same infrastructure. I think this is nice innovation. And those of you that have been visiting us the last half year have seen 4 Hamiltons. But just last week, we actually got 2 more. So now we have not 4, but 1, 2, 3, 4, 5, 6 in total. So we have 2 rooms with 3 in each room. And I was told, asking Hamilton that I asked how many robot systems are there in Europe? And they answered 40 in total. So we have 6. And there is 1 site in the world with more Hamiltons than us, and this is South Africa. The police department in South Africa have 10 Hamiltons. So we are at 2nd place now, and soon 1st. Yeah. So these 6 now, they serve 18 double GCs, and 18 double GCs are 36 single GCs, which is also quite a lot, So this kind of equals then with double capacity, right? Cutting the time in half, 72. Yeah. And this turns our capacity to 100K per month or 1.2 million per year in a very, very tight place. And we have some new innovation on the way as well. So this is our new GC. We just had 1 kind of test GC for a half a year now. We just ordered the 2nd one. And this one is quite nice. It's much smaller. It has double capacity of the other ones. So in just 1 by 1 square meter, we can have 4 of this with 1200 samples capacity, loading capacity. So yes, we are prepared for the future. And this is your numbers, start from the start, 2013, few samples, close to 200 in 2023. And I think I didn't actually update this slide, but I think you passed 250,000 samples in 2024. So with 1.2 million samples per year, I think we are good for some time, even with Hilde and Ørjan's kind of growth plans. Because if I extrapolate from the numbers, it will look something like this. Probably this is far too low, Hilde, right? Yeah.
We have big ambitions.
Yeah. But I guess I just want to show you that we are prepared, Bring it on.
Accelerate, Thomas. Trust
Hi,
us.
Ørjan.
Accelerate. Hi.
Hi. So with this slide set, following the credibility, I would like to show you that we do innovate. We don't rest. We innovate every day. Small things, refining the analysis, and especially the automation and the capacity, so we can keep costs down and also have sufficient capacity for the future. Then some slides about quality. And a common term for this would be the assay performance. How well do the test and the service perform? And to answer this, we need to define the purpose of the service or the test. This is quite key. Actually, it's impossible to validate or test something before you know the purpose. So using vitamin D as an example, what is the purpose of this test? Well, in my opinion, the purpose is to decide if people are in the low red region, being deficient, or if they have toxic levels of vitamin D in the blood. And it's kind of not to have a number of 65.2 or 65.7, so this is nitty-gritty. Because if you are at 15 or 18, you're still deficient, and if you are at 250 or 400, you still have toxic levels in your blood. And I think this is quite important, to not overpromise to the customers, because then it's so much easier to keep your promise. So a very important tool for us when we do validation and we have ongoing quality control during the testing and the analysis is quality control samples. And for this, we use dry blood spots. We buy a lot of blood from the blood bank at the hospital, maybe 20 liters. We screen the half-liter kind of blood bags from each person for high and low values, and we kind of put them into categories, low, medium, high. We mix them together and we make tens of thousands of dry blood spots. We put them in the freezer and we use this to monitor the precision in the accuracy. The first up is precision, meaning how similar are repeated analyses of the same sample. And this is expressed as something called relative standard deviation, which is a value with percentage. And this is a plot illustrating this. So you can see here in the middle, the blue, we have the Cincino samples, and before them and after them, we have these quality control samples. And since these are supposed to be identical, they should produce very, very similar results. And you can see these gray dots here, they are each QC sample results from each QC sample. And on the x-axis is time. And the green line in the middle here is kind of the average value, the true value, 100%. You can see they are not identical all on the green line, but they kind of deviate a little bit up and a little bit down from the average value. And this is the nature of any measurement, any test. You know, if you play dart and you throw the dart arrows, you will never always hit in the middle. You will have like a distribution around the middle. And this is what we see here. And if it look like this, we are happy. Sometimes we have some deviations like the one in the blue pulsing circle up here. And if you see something like this, we need to check the calculations. And if you find something wrong, we can correct this and post the results. If you don't find anything, we have to do a reanalysis, and this is why we need a second blood, drop of blood for another analysis. And this shows kind of the variation then for the fatty acid analysis. You can see this is C16, one of the highest concentration fatty acids. And you can see the CV, the variation is very, very low, very tight, less than 2%. Looking at one of the lower ones and important ones, EPA, you can see that the variation is somewhat higher, 7.1%. But it looks sound, you know, with a few outliers looking like these. Then the accuracy, and I think this is probably the most important one, but also the most difficult one to do. How well do the result compare to a true value? And there are a number of ways to kind of investigate this. One way, for instance, for vitamin D, is to kind of do a comparison between a plasma sample, the yellow fraction on the top of a whole blood sample, and the DBS sample that we have prepared from the whole blood. And then we do analysis in both sample types and we plot them, and you can see that, for instance, for a value of 100 in plasma, we have, after conversion, also a 100 value in the DBS sample. So this is one way. Another way is to use something called a standard reference material. And these are issued from kind of agencies. This is National Institute of Standards and Technology in the US. It is a kind of standardized freeze-dried plasma sample with true values of vitamin D. You can see here at level 1, the concentration is 71.8 nanomoles per liter. So by using this in our analysis, we can check if the results we get are accurate or not. Another way to do this is to join what is called external quality assurance programs or round-robin programs. And this one is for vitamin D. It's called DECAS. It's the largest one in the world. More than 1,000 labs from all around the world participate in this. And the coordinator, a UK university hospital in this case, then makes in the same way like we do the QC samples, like thousands of plasma samples. They are all identical. They have the kind of standard method, reference method to analyze the true value of the sample. They distribute the samples to all the labs in the world. The labs use their methods, their people, their machines to generate their result, send it back by email to the coordinator, and they compile this report where you can kind of see how your value compares to the true value. And if you are within the criteria, you have this diploma stating that you are fit to do this analysis in your lab. And we have been a part of this program since 2015, and we have been compliant every year. Another example is for HbA1c. In this case, this is a kind of diagnostic analysis, so it's a lot of tools out there to validate and compare your results with true values. And these are some value standard materials at low, medium, and high concentrations. And you can see that we use this and we analyzed using our method, and you can see this is the true value, this is what we found, and this is the accuracy, and this is also the variation in the analysis. So you can see it's very, very low, and the accuracy here, average 102, 106, is very, very good. We also did like an external comparison with the Swedish hospital. This is Sahlgrenska outside Gothenburg. And this is 264 samples spanning the whole range of HbA1c values sent to us blinded. We did DBS analysis from these samples, and this is on the other axis, and you can see that the regression equation here is extremely good. This is perfect. This R is 1, then it's perfect. So this is 0.969, which is really, really good. We also participate in an external quality assurance program called Equalis for HbA1c. So this is the true nominal value, 40.9. This is our result, very, very close to the nominal value. Another important thing to check is the stability. Of course, when you send samples from home in mail, you need to check the stability. So this is the stability for 3 different persons at 25 degrees in the sample bag. For 21 days. So you can see over time, there is no change. The same at 50 degrees, maybe a slight decrease, very small, over 21 days. For fatty acid analysis, we did it differently. We had 15 different persons that did a sample in the lab. We did the analysis after a few hours. Then we sent the sample in the mail to home to the participants. They mailed it back to the lab after some days. So the travel time was 7 days ambient, and then we kept it 3 days in the fridge when it kind of reached the lab to mimic the real situation. And you can see the blue and the orange analysis is comparison before and after mail shipment. So after doing all of these kind of test validation, then you are ready to say if the service is fit for purpose. And of course, in my opinion, and the data says that yes, it is. And this is it. And I'm 2 minutes before time.
Thank you so much, Thomas. It's a great presentation. Awesome job.